welcome everyone to today's webinar. We're kind of taking a little departure from what we normally do, which is usually talking about our instruments or interviewing or talking to researchers who've used our instruments. We're gonna kind of switch gears here and just do a little bit of a trivia session with you guys. And we're really excited to try this out for the first time. And thank you for being here and participating with us. Some of these questions are going to seem probably a little bit easier to some of you if you're familiar with plant science or agriculture, but it is going to span a pretty broad range of knowledge regarding everything from plant physiology all the way through post-harvest agriculture kind of applications, as well as a couple of questions about our company and a couple of questions about current research that has just come out this year. So we're going to have a lot of opportunities for people to demonstrate their knowledge of this entire industry. So for those of you, just for introductions, those of you who don't know already, my name is Galen. I am the Director of Applied Science at CID Bioscience and Felix Instruments. And with us joining today, who will also be helping ask some of these questions, is Susie Truitt. So Susie, go ahead and introduce yourself.
Yeah, my name is Susie Truitt, and I am the Director of Distribution and Operations here for CID, Felix, and now Interscan. My claim to fame is usually I'm the silent person on the webinar. So today I actually get to talk. So looking forward to this. How this is going to work is when we advance to the question, Galen or myself will read the question, and the timer kind of starts on that. The options will be down below on the slide, and I think you'll have another 5 seconds or 10 seconds to answer the question. The system also notes how quickly you answer the question. So in the event of ties, it's going to determine how quickly you answered your question. So the faster that you can answer the question, the better. All right, let's go.
All right, so let's get started. I'm going to close out that QR code now so we can get this full screen again. And Susie, you can do the first question, then we'll just go switch back and forth every other question.
Which of the following plants is a monocot?
All right, we got a bunch of people. We got some
smarty pants.
Yeah, get some experts with us. Obviously, sunflower and pea plant being dicots, pine tree being a coniferous plant, so it's neither a monocot or a dicot. So good answers from everyone. And then also will give us a leaderboard. So really quick answers from Tara and Arjun. So it looks like it's already going to be a tight race here for the first place. So question 2. Which of the following is not a primary plant macronutrient? Wow, you guys are quick. We didn't even have to use up the whole 10 seconds. All right, let's see how the leaderboard looks now. Oh, just a little bit quicker on that one for Tara. So, okay, looks good. You guys are doing great. This is really tight in the lead here. All right, ready for the next question?
Hopefully the questions are going to get more challenging then.
Yeah. All right, so the next question, Susie will read for you guys.
What is the function of root nodules in, oh my gosh, help me out.
Leguminous plants.
Thank you.
Legumes.
I was a history major.
All right, let's see.
Ooh.
Providing shelter to bacteria. Okay. Yep. So we've got some people thinking that the most primary function of the root nodules is to store food and nutrients, but the function of giving shelter to the functional bacteria that can fix nitrogen is actually the correct answer.
Oh, we had a leaderboard change.
Here we go. Changing things up. This is exciting. All right, next question. Question number 4. Which of the following is not true about mycorrhiza? Which of the following is not true about mycorrhiza? All right, everyone's answered. Correct. So this actually is a funny wording for this question. It's actually the reverse, right? The plant provides the sugar to the mycorrhiza, not the reverse, where the mycorrhiza are not there to provide nutrients or energy to the plant. It's reverse. So good job, everyone, on that one. Let's see how the leaderboard looks now. Oh, wow. We've got a big, strong lead now.
Ooh.
For Jerica.
All Jerica's right. Crushing it right now.
Let's see how the next question changes things up.
In crops, an increased leaf area generally leads to
increased light interception. A lot of you, yep, that's a great response. A lot of these other ones are just kind of completely false. So Jerica still holding on to that lead. Let's see if Tara, it looks like Tara's making a comeback though from her beginning early lead. So let's hope that we can see some comeback action here. So question 6. In agronomy, why is leaf area sometimes artificially increased in young crops? So in agronomy, why is leaf area sometimes artificially increased in young crops? To promote rapid canopy closure is the correct answer. So let's see where we're at now. Awesome. Oh, wow. There it is. There's that comeback. This is going to be a fun trivia to watch. Who comes out on top? Just so you guys know, there are multiple tiers of winners. We are awarding prizes for the top 3 contestants of today's trivia.
And no employees or CID of Felix can win a prize.
Correct.
Leaf area index can be used to study all of the following except
Ooh. So this is an interesting one. So a lot of you thinking that you can't really use leaf area index to study the effect and spread of pests and diseases, but in actuality, for disease counting and for looking at disease, you could use leaf area. You can also use it for looking at plant growth for abiotic stresses and changes. But you can't use leaf area to actually investigate direct photosynthetic efficiency. You can't, you can't actually know how efficient a photosystem is without doing some sort of gas exchange kind of measurements. So this is actually an interesting one where one person got that right. Looks like, oh, there we go. Arjun back in second place. So let's look at the next question. Is this me that I'm reading? I think it's me.
Yeah.
Which of the following is not a benefit of leaf spectroscopy measurements? Which of the following is not a benefit of leaf spectroscopy? Oh, interesting. Okay, so a lot of you okay. So another difficult question. So with this technology, spectroscopy in general, especially when you're talking about NIR spectroscopy, being able to quantitate trace chemicals is something that technology very much struggles with. Trace chemicals are not, because they're in such low concentrations, not easy to detect with NIR technology. However, there's a lot of research out there where people are using spectroscopy to help monitor for early disease detection. Small changes in the invisible, the non-visible light spectra, and even some in the visible, when you're looking at chlorophylls and carotenoids and other photopigments, are being used to, those changes are being used to look at early disease detection for things like HLB in citrus trees and other disease detection applications. So correct answer there was trace chemicals. Cannot do that.
Ooh.
All right. So leaderboard's looking pretty stable still here. Let's move on to agriculture questions.
Oh, here we go.
So question 9, Susie.
Which component in fruit has the strongest absorption in the short wave near infrared region of light?
There we go. That's what I like to see. Some people that know their spectroscopy. Yes. So water is the most strongest absorbing component, mostly because the OH bonds that are being vibrated in near infrared spectroscopy, which is vibrational spectroscopy, have very strong overtones in the near infrared region. And so they are, water being comprised of strictly OH bonds is the component that is going to be most easily detected using near infrared. So let's see where we're at now. Looks like we're getting some people catching up to Jerica. So I think this next question is going to be pretty big.
Very consequential.
So question 10. Which fruit is not an example of a climacteric fruit? Which fruit is not an example of a climacteric fruit? Watermelon. Watermelon is not a climacteric fruit. All these other ones are climacteric fruits.
What makes something a climacteric fruit?
So, sorry, Susie, I can't hear you. What was that?
I said, what makes something a climacteric fruit?
That's actually a great question that we should pose to the chat. Somebody want to define climacteric fruit for us? Who got the answer correct, I guess, probably.
Like I said, I was a history major. We didn't study that at all. Although I did go to school at the University of California, Davis, which is one of the big ag schools in the world.
Whoops, I'm going to go back.
Hold on.
Oh, no. I think I just messed something up.
I don't think you did.
Okay.
I think we just went back to
hopefully not. I didn't mean to. I forgot that it automatically advances to the question.
Yeah.
I was just trying to get out of the leaderboard. So, so we're going to the next question here.
We're going to the next question, gang.
All right. And this one is for, oh, geez. I did mess something up. Oh, no, there it is.
Oh, that's cool.
That's you, Susie, right?
Yeah. At which of the below ranges of dry matter content are Hass avocados generally considered to be mature enough to harvest?
So which dry matter range are Hass avocados mature enough to harvest? That was a quick one. That was not a lot of time. But yeah, so this one, this is an interesting question because we do kind of butt up right on that line of 20%. But generally, most regions, the regulations are between 20 and 25% for the dry matter content before they are allowed to be harvested. And so that's why 20 to 25% is the correct answer. Some regions might go as low as 20%, maybe even 19.5. But for the majority of the larger avocado growing regions, it's going to be between 20 and 25%. 25% usually being on the high end. It's usually more closer to 20, between 20 and 22. Oh, no big changes in the leaderboard. Miriam coming back up. So next question. Which of the following fruits does not commonly use dry matter content as an indicator of maturity and quality? So which fruit does not typically use dry matter as an indicator of maturity and quality? There we go, table grape.
There we go.
Yep. So table grape typically using things like bricks or titratable acidity, color even, pH, things like that are more common for table grapes as well as other small berry fruits. So let's see how things are going now. Oh, and Tara's back up in the second.
And Ken's climbing the ladder.
Yeah, Ken's climbing the ladder as well. All right. Someone's got to dethrone Jerica though. She's Jerica's been holding the lead for most of the trivia here. Who's willing to dethrone the current champion? So let's go to the next questions, which are more related to post harvest. So maybe Jerica doesn't know much about post harvest. Maybe we can maybe someone's going to get an opportunity here. So let's start with the next question. This one Susie will read out loud for you guys.
Which of the following is not a benefit of controlled atmosphere storage of apples?
Awesome.
Oh, there we go.
Yeah, good to see you guys know your stuff. Controlled atmosphere actually has been shown to actually kind of decrease flavor in a lot of studies. So yeah, that's why controlled atmosphere is not a flavor enhancer, but it does do all the other things. It does inhibit growth of microorganisms, slows ripening, helps maintain the firmness of the fruit if you're at the right controlled atmosphere settings.
And means we have fruits and vegetables available year round.
Yes. So now we have. Okay. This is gonna be an interesting.
Ken dropped off.
Ken dropped off, but we still got 6 or 7 questions left. So we should be. And there's a couple in there that I think might throw some people off. So hopefully we'll see. Start with the next question here. Question 14. Which gas, if accumulated in high enough concentrations, can cause stored crops to begin ripening in controlled atmosphere storage? So which gas, if accumulated in high enough concentrations, can cause early ripening in controlled atmosphere storage? And this was a pretty easy one. I was hoping that we would get everyone on to agree on this one, that ethylene, the ripening hormone, is the gas that is going to cause early ripening. And so that's why in controlled atmosphere, we try to control ethylene levels to make sure that fruit don't ripen too early.
And that's why we make instruments that measure that.
As well, yes, so we can monitor our ethylene levels. Those are some quick answers. 96, I feel like that's within 0.04 seconds of the question being able to be answered. That's amazing. So good stuff. Longest streak right here for Jerica.
3 in a row.
Question 15. So besides scrubbing ethylene from the environment, what other measures can be implemented to prevent the ripening of stored fruit in controlled atmosphere storage while still maintaining quality? So besides ethylene scrubbing, what else can you do to make sure that the fruit doesn't ripen? So this is pretty even spread here. So all of these other things. So I think the caveat here that was hard. This is the longer question. We probably should have given a little more time on that to be fair. But the caveat on this question was while still maintaining quality. And so things like changing the actual parameters of your controlled atmosphere storage, for the most part, if you decrease temperature more than you're below your limit, you're going to decrease fruit quality. If you reduce your humidity, you could also risk reducing your fruit quality. Same with overly saturated CO2. You could risk reducing that quality of that fruit. But 1-MCP is a newer kind of technology, or I guess chemical being used, chemical treatment being used to help block ethylene from being able to be received by the receptors on the fruit, and therefore reducing the potential for it to ripen artificially, even if exposed to trace levels of ethylene. And so that's why that is the correct answer. That doesn't have any impact on the quality of the fruit, doesn't change or control the atmospheric conditions necessarily. It just blocks the ethylene. So, Susie, I'll let you read the next question, and then we'll get back to our back and forth.
No problem.
So, let's look at the leaderboard first. All right, no changes in the
Jericho streak.
Top 3.
Lost on that one,
yep.
But still at the top of the leaderboard.
So now we're getting into some questions that are going to be related to new research. There's only, I think, a couple of these, but these are new discoveries from 2023. And so this comes just directly from research that I've read this year, and I found to be quite interesting and related to plant science and agriculture. So,
and I don't think any of them were on TikTok, so I probably won't know the answers.
All right, so Susie will read this next question for you guys, but we're getting close to the end, so make sure you're getting these answers right and as fast as you can.
3 new cultivars of which crop were recently released by the USDA ARS and are the first cultivars of their kind designed for both human consumption and planting in the fall?
Wow, nobody guessed pea.
Right.
Yeah, so if you guys I don't know if you're subscribed or not, but the USDA ARS releases a new discoveries kind of I guess almost like a newsletter, but it's a big briefing of all their different discoveries. And they just recently released these 3 new cultivars for pea here's the caveat, they're designed for human consumption. So they do have fall planting pea varieties, but those are usually meant for feed, for livestock. But these ones were designed specifically for human consumption and can be planted in the fall. So they can resist those winter conditions. So interesting here. I mean, yeah. I put the main 3 crops in there because I knew that would throw some people off to see all of them there. So yeah, that's some interesting new research for you guys. So since no one got that right, leaderboard does not change. And the next question is also about new research. So this is question 17. A recent study from the University of California, Riverside demonstrated that which of the following degrades the service that beneficial soil bacteria provide to roots? So this study showed that what of the following degrades the ability for mycorrhizal bacteria to provide nitrogen fixation? And the correct answer, 3 people got it. Competition between strains of rhizobia. Now, these other things that I put on here are things that can degrade the service of beneficial soil bacteria. So I did purposefully make sure that there was all these things that could also potentially degrade that service. But because this is referring to this recent study, this is what the recent study was actually investigating, was this competition between strains of beneficial rhizobia. And they're basically looking at native species, native strains that are already in the soil, and then lab grown, highly beneficial, genetically engineered strains of rhizobia, putting them in there, and always finding that the natural rhizobia that were less efficient at fixing back nitrogen were always outcompeting the really beneficial lab grown strains. So which puts some interesting research into perspective as far as treatments that we've been looking at in ag chem and biotech kind of solutions for fertilization, where we're at, people are wanting to add in these beneficial soil microbes, but it turns out that this competition is pretty stiff, and it's something that we're going to have to figure out how to overcome if we're going to be able to use these alternatives to classic fertilizers. So those were two studies that I really enjoyed reading. And so if you guys have a chance, I would, for both of those, I would go and look through and read up on those, because they were both pretty interesting discoveries, I thought. So the next questions, well, actually, we'll look at the leaderboard here. Oh, we got some no shake up in the top three. No shake up in the top three at all. Okay, so we've got only, I think, three more questions left, or four more questions left. So these are going to be related to the company, I believe. So these are questions about CID, Felix Instruments, Interscan. And those questions, this, I feel like, kind of evens the playing field a little bit, because it's not so much reliant on your background and your education, but more so on your knowledge of just who we are and how we operate. So let's go ahead. Susie, you can read this next question about us.
All right.
This is question 18.
Put the following products in chronological order of release date.
So what we're looking for is to make sure.
Which one was released first, which one was released last, and put them in order.
Oh, we had one person get it right. Wow.
All right.
I'm impressed. That was a tough one. I had a feeling that this was going to be the one that no one was going to get. But yeah, this is exactly correct. So good job. I wonder if it'll show us who got that one right. Let's see if I can scroll down. I want to see. I want to award. Oh, Hunter. So no one did actually get that right today. Yeah, that's a tough one. All right, question 19. This is another company question. What year was CID Bioscience founded? What year were we founded?
So this is just a good guess, guys.
I have a feeling that we'll get maybe 2 to 3 people will get this right.
We'll see how many people need to play the lottery today because they made a good guess.
Wow. Impressed.
There we go.
All right. 4 people guessing correctly, 1989. Let's see here. Oh, wow. Last minute shift for Arjun to get up to second.
Could we have a complete upset with the last question?
We have one more question. It's, I mean, the most you can get, the most I've seen anyone get is 96, I think. So I think 100 you would have to answer before, like, the instant the question becomes available to be answered. So I think it's still possible, if Jerica does not get this next question right, Arjun could potentially take the lead. But let's find out on this last question. This one's all you, Susie.
In which city is CID Bioscience headquartered?
Let's see how fast you guys can Google.
And while you're Googling, we would invite you to visit us in our fair city if you are ever in the area. We are right on the edge of the Columbia River Gorge. Oh, there we go.
6 people correct. Camas, Washington.
Camas, Washington, home of the paper makers. Our city was founded with a paper mill, which still operates.
And it's a wonderful city to visit. The Pacific Northwest is beautiful. Highly recommend.
And we really do invite anyone, if you are ever in the area.
Oh my gosh, the upset, it happened.
It happened. Holy mackerel, Arjun. That's holy
incredible. I did not think that was gonna happen. Wow.
Good Jerica, for you. That was a
you put up a great fight.
That was amazing.
Wow. Yeah, that is
One
quite a
of the best
comeback.
last minute comebacks. Yes.
Well, congratulations, Arjun. Jerica, honestly, you did amazing. You answered most things correctly. That was amazing. And Tara came back from uh, the early lead and then dropped off the leaderboard and then also came back to get into third place here. So Arjun, Jerica, and Tara, you guys are all the winners today. And we will the way this will work is since you had to enter your email for the AHA slides, we now will be able to just email you directly about your prize. So congratulations again. We want to thank everyone. This is just a kind of a fun way to explore plant physiology, agriculture, post-harvest questions. Everything that we talked about has to do with parameters for instruments that we create that can measure those specific kinds of parameters. So we just wanted to do something fun for you guys. It was fun for us as well. I had a lot of fun doing this. So we probably will be doing more of these in the future, I would think. So thank you guys all for joining us today. Thank you everyone for your participation, for answering these questions. If you have suggestions on how we can improve this, I already have one off the top of my head. I think that we need to increase the time just slightly for some of those longer questions. But other than that, yeah.
Thanks so much for joining us today, you guys. And again, as I said, we would welcome anyone if you're ever in the Pacific Northwest area. Camas, for those of you who haven't looked us up on the map, is just right across the Columbia River from Portland, Oregon. And we engineer and manufacture all of our instruments here. And so we'd be happy to show you around our facility if you're ever in the area.
All right. Well, with that being said, Arjun, Jerica, and Tara, congrats one more time. And we will reach out to you so that we can get you your prizes. And for everyone else, come back next time that we do this and give yourself another opportunity to win some money. So, all right. Thank you, everyone.